So I'm very excited to introduce Professor Ross Young as our third keynote speaker for the conference. Professor Young was a very exciting lecturer as someone who studied under him at the University of Queensland too long ago to remember. So I'm excited to hear him speak again. Long since he's left UQU, Professor Young was appointed the Executive Dean at the Faculty of Health of QUT in January 13. That was following 7 years as an Executive Director of the Institute of Health and Biomedical Innovation. It's not going to work with me being on a loudspeaker. Take her outside. Professor Young is a clinical psychologist and his research interests lie in the integration of psychological and biological risk factors in mental illness. His research includes work on substance misuse, schizophrenia, anxiety disorders, mood disorders, and more broadly in behavioural medicine. This includes work in pharmacogenetics and personalised medicine. Professor Young has over 250 published book chapters and papers in generic medical, psychiatric and psychological journals, and he's a member of many boards, including the chairman of Mental Housing, which is a non-for-profit organisation building housing for those with enduring mental illness. So please join me in welcoming Professor Ross Young to the stage.
Thank you, Julia. Lovely to see you again and very nice to be in Perth. Look, to get things going, I'm going to share with you one of the most important academic volumes of 2015. It's called Is Gwyneth Paltrow Wrong About Everything? And the answer to that pretty much is yes. It draws attention to issues of populism, despite evidence, or ignoring evidence because it challenges our stereotypes in an inconvenient way, and talks about the culture of celebrity and the legitimacy of culture of celebrity in terms of informing public opinion. Now, Gwyneth, as you may know, she discovered healthy eating, and that healthy eating is the fast track to good mental health. Apparently she had a lot of unexpressed anger. I think that just means she was bonkers. And the things she recommends to transform your mental and physical health are things like kale. It's never anything cheap or available, is it? It's never cabbage. It's got to be kale. And wild blueberries, not the toxic farm variety, but the very expensive wild variety. And on her website, which is a truly awful piece of social media, it's called Goop, Dr. Oscar Sewellak, who, you won't believe this, he's from Nimbin in New South Wales. So he's an international authority on lots and lots of things. He talks about something called postnatal depletion, which, Julia, you should be really concerned about because it can last for 10 years after you've had a baby and is related to low B12, B line and selenium levels. So Gwyneth's got a lot of material here, but almost none of it has any scientific evidence. The most important theme of this book, though, is that our knowledge is bound by culture, whether that be celebrity or culture or any other kind of culture, including in science. And in the addictions, we often have a clash between those that are driven by more biomedical and genetic views of the world and those that are driven by more social or environmental views of the world. And the relative merits of nature versus nurture have been discussed for a long time in terms of our discourse and scientific history. Someone called Whit first wrote about it in 1765. But I'm going to talk about our work and its attempts to unite those perspectives because they're both important. And rather than be in a world of reductionism, we need to be in a world of integration where we're building on our understanding in an integrated and sophisticated way. And so I hope that's what we'll begin to explore today with work that we've been doing for many decades. And this is work that I've undertaken with my colleague Bruce Lawford, who's here, my good mate Bruce, and our colleague Ernest Noble, who's at UCLA. And stands counter to some of the debate that we see, particularly driven by the social sciences, which talks about things like a neuromolecular gaze or neuroreductionism, and is often very critical of those approaches in terms of their translational potential for things at a population level or public health. So today there'll be three main themes that we're going to look at. One's a look at substance use with a particular focus on dopamine, and particularly the D2 dopamine receptor gene, which we've looked at for over 2 decades now. And then examining how we can understand, using the models that we've used a greater window on complexity, on substance misuse, on response to treatment, and on comorbidity in particular, and I'll draw mostly on our work in post-traumatic stress disorder there. And then lastly, offer the promise of epigenetics, and that won't be based on data that we have to date, but data that we're currently collecting, but something that I think really has the potential to build a bridge between the social and environmental and the biological and genetic. To begin to explore some of those issues regarding the underlying mechanisms of addiction, and Wayne Hall and his group at the University of Queensland have been exploring popular models of addiction, and in particular using the frame of reference of the brain disease and how that's become widely accepted internationally, but one that Wayne and his team thinks has shifted our view away from those more public health and preventive approaches. But this is some data that the team have published this year, and it's looking at someone who has a family member with a substance misuse disorder and their attribution of what the causes, and there can be more than one cause, but what the causes of those problems might be. And so if we're examining populist notions, we can see that there are things like 82% agreed that the enjoyment they got out of taking drugs was what was instrumental in terms of the family member developing addiction. Problems with coping, poor self-esteem, personality, easy access, but genetic makeup features there too, and chemistry in the brain, and risk takers. So if you look at the kind of popular notions of what might be driving addiction, and particularly something that someone will be finely tuned to if they've got a family member with a substance misuse problem, you can see both the biological and the social featuring in terms of those lay descriptions. Our own work began, grounded in psychological theory, looking at the work of Bandura, and typically used a social psychological approach. So we were very interested in the formation of alcohol expectancies, of beliefs about the reinforcing potential of alcohol, and how they might be mediated through your capacity to be confident in resisting alcohol and subsequently result in alcohol-related problems. And we've got lots of data that supports that model, and we've got cross-sectional prospective data with several drugs of abuse as well, including alcohol, cannabis, amphetamines, etc. And they're also predictive over time. So this work that we've done with Jason Connor, who undertook his PhD with me, looks at young people and the development of their drinking, and whether the beliefs they hold about the reinforcing potential of alcohol and their capacity to resist alcohol in the face of social pressure or feeling sad, predicting the emergence of problems over time, and it certainly does, and we've replicated that in several subsequent studies as well. So the essence of the model seemed to be pretty good, except we were also intrigued by what might be driving these beliefs. How do they arise? Certainly they arise out of things like observation of members of your family, that they arise from media influences, from our broader societal and cultural values, but they also arise from how we're hardwired. And so we were interested in bringing genetic research to the equation to try and inform how beliefs are formed and their potential influence on subsequent behaviour. And just before we begin that little journey, I'll give you some caveats that need to be heeded as we think about this body of work. One, that much of the work that exists in the genetics of addiction, as well as the genetics of many other mental illnesses, hasn't been well replicated. And science, the essence of science is replication, and so that's just something to be aware of. We need to avoid biological reductionism, and the epigenetics argument I'll put to you later on will certainly put that case in point very strongly. But so just because we're talking about genes doesn't mean that these genes operate independently of environments. Both are vital to consider as part of the equation. And we've also got to look beyond association studies that just compare diagnosis A with no diagnosis, but to drill down into what the functional outcomes, what the broader symptom clusters might be that relates to these environmental and genetic processes, and where possible consider multiple genes. And that's an emerging body of work, how we deal with that statistically, how we look at gene panels and their influence on behaviour subsequently. And not to forget that measuring environmental issues such as stress, environmental pressure is a vital part of the equation, and to look at how these collectively operate over time. So much of the work's cross-sectional and not longitudinal, and it's very important that we look at how these cascade, particularly during crucial developmental epochs. And lastly, and I'm only going to mention this briefly, but that's in no way related to its importance, that this broader body of work in terms of genetic research has very vital ethical implications, particularly as we consider issues of access to care, of stigma, of insurance, of potentially job-related discrimination. So there's a broad ethical framework that we need to consider this genetic work in as well, and that's including people's own sense of identity if they've been identified with genes that are high risk for a particular condition. These aren't just really cute animals, they're called prairie voles. Microtus ochrogasta, and they've been well studied in terms of their brain circuitry and dopamine. And dopamine is crucial to the formation of a pair bond in these cute little animals. Only 5% of mammals form lifelong pair bonds, and these little guys are one of that 5%. And their brain circuitry's been very well studied in terms of how they form these social affiliative bonds, and dopamine is absolutely crucial to that process. Drawing on this body of work more broadly, the value of dopamine in multiple studies has been seen as alerting the organism to new or novel stimuli in the internal and external environment. And it's very important in terms of us improving our survival value, our adaptation to a whole range of environmental forces, including social and affiliative bonds. And that's a very powerful evolutionary advantage, that we're working together as social beings and networks to look after our collective survival. And so when we look down the phylogenetic chain, we can see that dopamine or its precursors plays a really important role in terms of our adaptation and our capacity to survive. We also see that in human studies as well, and this is one of Nora Volkow's findings from her numerous studies looking at brain imaging, but essentially painting a very robust hypothesis, largely based on work using methylphenidate or stimulants that shows the relationship between low D2 dopamine receptors and liking or reinforcement that relates to drugs. So there's a very good body of human work that also underscores the importance of dopamine and brain circuitry in the addictions, building on this broader evolutionary perspective that we've seen in animals such as the prairie vole. And so when we begin to coalesce this together, and this is a recent study that's begun to look at a whole range of genes, and using a really sophisticated approach in Bayesian statistics, which is called Convergent functional genomics. And essentially what this group's been doing is to try and build a picture of all of the extant data that we have, whether it be animal or human, looking across a whole range of different markers to try and identify risk that in this case would relate to addiction. So they've used something called genome-wide association studies. I know most of you will probably heard of them, but they're essentially looking at the whole of the human genome and comparing a group with an identified disorder and those without. So they're not hypothesis testing entities. The genome-wide association study is just looking for naturally occurring differences, usually has very large sample sizes, but often those samples aren't very well described. So the phenotypes are quite thinly described because you've got to recruit 10 or 1,000 or more people in your study. So while they're useful, we need to recognise some of the limitations of those studies. So the great thing about this convergent functional genomics approach is that it brings together not just those studies which are large and not hypothesis-driven, it brings together that with hypothesis-driven association studies as well, and gene expression data, whether they be for animal or human studies. And they've conducted these pathway analyses to try and identify which genes might be most important. They've done this on the basis of creating a panel. So that panel was 135 genes, and within that 713 markers of single nucleotide polymorphisms, and particularly focused on alcohol dependence in the data I'll talk about today, and have large numbers of cases. And these are the top candidate genes that they've found in terms of all of this work bringing it together, including an animal model. And the first one listed there, the synuclein alpha, interacts with fat and forms presynaptic vesicles that relate to dopamine release. The second, the glial fibrillary acidic protein, relates to astrocyte formation in the brain. And the third one is the D2 dopamine receptor gene cluster that I'm just going to talk about in some more detail. But that was the third most important candidate gene that was identified through this very important body of work. And when they looked at the interaction of these genes with several other extant bodies of data, the DRD2, which is what I'm going to be talking most about today, is of interest because it overlaps between anxiety and schizophrenia and addiction, and also this animal model as well. So while there are numerous, these are the genes that have identified across these different studies as being important in this integrated body of work, but I'm going to focus on this one most because we've got more data on this than anything else, and also because through this very extensive process, it appears to be the most relevant marker that we have in terms of comorbidity, which is a particular theme to explore today. But just before we begin that, to acknowledge that there are multiple pathways to all disorders and that none of the data I'm going to talk about today are causal. So while we're introducing the notion of this gene as a risk, or other genes as risk factors, not to overstretch in terms of our interpretations of this. But certainly the DRD2, the D2 dopamine receptor gene, or its adjacent gene, the ANK1, which is a kinase gene sitting alongside it on the long arm of chromosome 11, have been widely studied in terms of addiction. The ANK1 gene relates to signal transduction and probably modifies the expression of that D2 dopamine receptor gene, influencing the number of dopamine receptors and potentially their activity in the central nervous system. Blum and his team, including our colleague Ernest Noble, initially considered this to be in terms of reward, but we've now got a much richer and broader view of dopamine in terms of multiple functions in the brain, relating to motivation, to memory, to mood, and to executive function. So it's not just a narrow focus on reward. We need to extrapolate to a whole range of different brain subsystems for which dopamine is vital. And we also need to be reminded that we've got a stronger body of evidence in terms of the impacts of drugs of misuse on dopamine release and stimulation in the brain from work looking at psychostimulants and from alcohol than for other drugs. But the model's been applied to multiple drugs of abuse, but the data are strongest in terms of stimulants and alcohol. And we've conducted meta-analyses looking at this particular gene ourselves. Some of them have said that more recently that there's a possible publication bias. That means that this isn't as robust a body of work as it might be. But the largest meta-analysis to date, looking at this DRD2 and ANK1 cluster of genes on chromosome 11, has been published in 2013. And it's a meta-analysis of studies that include over 18,000 participants and in 61 case control studies. So it's a very robust body of work. And what it shows, and this is just looking at how you plot publication bias. So this is the pooled effect size of the different studies. And what this tail here shows from about 1999 onwards in terms of a publication year is that the publication bias isn't evident in these data, that the effect size is now stable, and this doesn't indicate that previous thoughts that maybe this body of work was biased and that the things that weren't showing an effect, the studies that weren't showing an effect weren't being published. This argues against that. So we can be confident that the body of work included in that meta-analysis is reasonable. Now we've conducted a number of different sub-studies to try and unpack what this gene might mean. And this is just one of these, and it really, it's a study of 72 healthy adults that we recruited from a community college or TAFE, and we subjected them to acute psychological stress, which was in the preparation of a speech versus a relaxation condition. And in fact, in the data that I'll show you today, the stress condition didn't make any effect. But we were very keen to look at the interaction between this environmental stress and a risky gene, and how you might approach new or novel stimuli, consistent with that model I've talked about before, that dopamine's vitally important in terms of orienting us to new or novel stimuli. And we did that through a number of psychological tasks. And Melanie White, in her PhD looked at this in terms of whether there was the carrot, which is a commonly used test looking at reward sensitivity. It's got 4 trials of a card task with small cash rewards. There was a delayed discounting task, which was a 2-choice impulsivity paradigm, and then a response inhibition task, which was a go-stop type task. So see if you suppress a prepotent response. And these also relate to different brain regions. So the response inhibition task is said to tap orbitofrontal activity, and the other 2 tasks, ventral striatal activity. But essentially are a very good way of looking at how people approach a task for new or novel stimuli, and we were very interested to see if this was related to genetic status in terms of the risky version of the ANK1 or DRD2 that we've been looking at, the A1 plus version. And essentially what this shows is that independent of stress, those with the A1 version of this DRD2 gene, particularly when the presentations were were short presentations in terms of microseconds, that they had a poorer inhibition at these very fast presentations, and their stop latency was also impaired across all of these different presentation times from 150 to 350 milliseconds in this go-stop task. So essentially what this was showing us, that those with this gene who hadn't yet started to drink were young people, but they had a more rash and impulsive approach to a task involving a prepotent response that we were trying to get them to stop. Our colleagues in the US have taken this one step further and looked at brain imaging studies, and this is Edith London and Ernest Noble's work, and they're also looking at young people without alcohol problems and seeing whether the presence or absence of this risky version of the ANK1 gene, the A1 plus version, is associated with both self-reports of mood and brain activation, and they administered 0.75 milligrams per kilogram of ethanol to these subjects or a placebo. And what they found in their studies was that those with the A1 plus version of this DRD2 gene reported less anxiety and fatigue, and they also showed increased striatal and insular activity. And this very much parallels what we heard from Sandra Brown this morning in terms of young people and the impacts that alcohol might have on them. So from these 2 bodies of work, not looking at clinical subjects, we concluded that there appeared to be a more impulsive group that had this risky marker, the A1 version of the ANK1 or DRD2, and that when you administered alcohol to this group, they responded differently than those without the risky gene. Now, this makes obvious then implications in terms of how we think about our clinical work, and so the data that I'll now present extends this basic work to looking at the clinical implications of these findings. And the first of the points I'd like to make is a really basic one, that the proportion of the group that you recruit who'll have this risky marker depends on where you recruit them from. And so Group A in this paper that we published in 1997 were recruited on the basis of presenting for acute detoxification. And as you can see, that about 15% of them had the A1 allele. Group B was recruited from a chronic rehabilitation unit, and we've got a higher percentage there of about 20%. And Group C weren't presenting for help with their alcohol problem, but clearly had an alcohol dependence and comorbid physical problems, and we recruited them from a gastroenterology unit, and they had an even higher proportion with the A1 allele. And conversely, when we looked at controls, we could see that for on the left, Group D, they were controls whom they had a family history of alcohol or substance misuse and had consumed alcohol in a hazardous manner in the past. Group E were those that had no family history of alcohol or substance misuse problems in the past. Group F were those who had reported no hazardous past alcohol use, and Group G were those that had no past hazardous use and no family history. And so we were intrigued by these findings, that many of the works that had been conducted previously hadn't really drilled down in terms of those they'd recruited as controls and asked them about their family history or their own substance use patterns. And certainly most of the studies hadn't recruited patients from gastroenterology wards. And in fact, many of the studies had excluded people with significant physical comorbidity. So we realised that the effect size you could obtain in a study was highly dependent upon how well you knew your subject pool and where you recruited them from. And we found that very interesting. We've proceeded to look at a number of independent samples. This one here, just looking at the relationship between this risky status and consumption. And once again, these are compelling results in our case. That we always uncode the genetic status last, and we've had time after time striking effects. So in terms of those with the risky version here of the gene, they're smoking on average 36.2 cigarettes a day versus 27. That their quantity in terms of standard drinks per day is 21.7 for the A1 plus group, 16.69 for the A1 minus group. And these people, all people presenting for help, for assistance to an alcohol treatment centre. But even amongst that population, we could see genetic effects. And then when we try and disentangle that in terms of the expectancies I talked about before, we see here the difference in terms of their self-report of why alcohol might be reinforcing or their self-efficacy in resisting alcohol. It was social pressure. And that was the biggest difference between the A1 plus and the A1 minus, with much less confidence in resisting alcohol under social pressure circumstances, which we thought was intriguing given that we'd already seen in animal models such as the prairie vole the importance of dopamine in social affiliation. So in terms of people's self-report and cognitions that they were reporting, we could see that there was an association with these genes. And this just looks at structural equation modelling that begins to draw this together with 143 patients, showing significant paths that exist from your allelic status through the age of onset of your problem drinking, with an earlier age of onset for those with the A1+, a direct association with drinking quantity, and an indirect association there with self-efficacy and expectancy via age of problem drinking. But this was tying things together in an intriguing and neat way, and something that we found compelling through multiple samples. I'll just go on a little bit of a sidetrack because we've also looked at a number of other markers. And we've also looked at a number of other populations. And just to prove that this isn't all about dopamine and alcohol, this one here relates to distrobevin binding protein 1, which is associated, a gene associated with glutamate regulation and, of course, excitatory damage in the central nervous system and the brain. And this is a gene that we've compared its association across a number of different diagnostic groups. I've listed here in terms of those with opiate dependence, PTSD, alcohol dependence or nicotine. And as you'll quickly note, looking at the mean ages in those groups, that we haven't matched them for age. That wasn't possible, so that's a confound in terms of what we're doing. But increasingly what we're finding with these broad genes here that relate to a number of different aspects of brain function, in this case potential widespread damage that's due to glutamate excitation, we can see that these are odds ratios. These are the genotype counts from the various genes, but we can see a significant genotype effect for schizophrenia, for post-traumatic stress disorder, for nicotine dependence and opiate dependence, and not, there's a trend here for alcohol dependence in the allelic analysis, but not for alcohol dependence. And they show two patterns here, a recessive inheritance pattern for opiate dependence and for schizophrenia, and a partial recessive pattern for PTSD and for nicotine dependence. And I'm only showing you that just to say that as we're progressing into this body of work more thoroughly, we're moving beyond just looking at one disorder and also trying to discern when genes might relate to one particular condition or where they might be related to broad changes in brain functioning that may interact with the environment to produce specific risks for specific disorders. And another important example of that's looking at comorbidity. This recent study that we looked at, we were particularly interested in brain-derived neurotrophic factor and its genetic regulation, because we know that that has profound impacts in terms of learning and of dopamine regulation as well. And we looked at 2 variants of brain-derived, the gene that is responsible for brain-derived neurotrophic factor, one RS6265, which we know is a functional gene that alters the secretion of mature brain-derived neurotrophic factor, and the other one is RS7103411, which is very catchy, but we know that in a previous study that's been associated with comorbid alcohol dependence and bipolar disorder. Now, why we were keen to look at this, because there's lots of evidence that suggests that BDNF's associated with the richness of dendritic carburization, of neural connections. It's related to learning. It's very powerfully affecting hippocampal neuronal connections and richness as well. So it's a very important complex to look at in terms of learning and memory. And in terms of this study, we had several samples, so we could replicate the study within the same replicate these samples within the same study, and this one came from the Australian Schizophrenia Research Bank, which is a genetic bank we could obtain DNA from as well. And we also had an alcohol dependence group and a control group with no diagnosis, who also came from the ASRB. And what we could see in this broad array of work was that alcohol dependence in schizophrenia was associated with brain-derived neurotrophic factor, but not an alcohol dependence alone. So it was the comorbidity, the self-medication with alcohol appeared to be a particular risk related to BDNF, and was also associated very strongly, so these are the risky versions of these 2 genes, the CC genotype and the AA genotype for these 2 BDNF genes we looked at. And in particular, we noticed a very strong effect in terms of impulsivity or risk-taking behaviour under the influence of alcohol as well, causing injury to self or others. So once again, it's a different level of sophistication by saying we're not just looking at genes that might either differentiate or identify common risks prevailing across groups, but it's also trying to understand comorbidity in a more finely grained way as well. So in terms of these cautious conclusions regarding genetic risk, that traits related to addiction are influenced by multiple genes, but in our own body of work, the DRD2 or the ANK1's shown significant convergence across studies, and particularly related to factors such as underlying rash impulsiveness, an early onset of drinking, particular brain activation, and lower confidence in resisting alcohol when reported social pressure. And more recently, we've been trying to move beyond these individual associations by understanding comorbid risk and developing gene panels as well. And we think that this is timely because clearly we're only doing this because we want genetic risk to be able to assist with the development of more effective and targeted treatments. And the NH and MRCs provided a genetic testing statement in 2011 talking about the identified clinical need for genetic testing, and it's going to be on our doorstep very soon. This is the Spartan RX, and it's a desktop DNA analysis tool that talks about DNA on demand, and it's particularly related to the metabolism of cardiovascular medications and cytochrome P450 activity. But it's allowing to understand groups that are fast metabolisers or slow metabolisers, and then to prescribe a certain dose of medications. But these are things that just a cheek swab and then a rapid DNA analysis within minutes can help to provide personalised medicine for cardiovascular disease. We've also seen over 500,000 consumers sign up for 23andMe. The FDA in 2013 said that if 23andMe was a medical device, and so they can no longer offer advice on health risk, so it's more about which part of the world you're more likely to have come from. But that's clearly something that's on their radar. This figure here is from a study called The Impact of Personal Genomics, which had 1,800 participants, and in terms of that looked at the advice that people or changes in behaviour that people subsequently undertook after receiving genetic advice, and 42% made changes in terms of their health behaviours. A similar study showing that changes were more likely if one had chronic disease, a family history of disease, poor health, or visited the doctor frequently. And there are industries now emerging in the US around this too. In fact, the US Institute of Medicine has just had a 2015 Genomics-enabled learning healthcare systems roundtable. It's a bit of a mouthful. This company here called GNS Healthcare is bringing together issues such as electronic health records and genetic information to try and make individualised predictions in terms of health. And there's a very large study at Brigham and Women's Hospital and Harvard called MedSeq looking at cardiomyopathy and how GWAS and other genetic data can help to inform clinical decision making. We're a long way off that in addiction, and I'll describe to you some of the simple findings that we've had in our body of work as well. One of these was a study that Bruce and I did that looked at response to methadone and over a 12-month period who responded well and who didn't respond as well. Of this group, 91.6% were using heroin daily when we saw them, but the A1 plus group were using twice the dose of heroin per day than the A1 minus group. And when we look at their outcomes over that 12-month period, we also see that at the end of the 12-month period, even within that poor treatment outcome group that I've mentioned here, those with the A1 plus status were using 0.63 grams of heroin per day versus 0.25 grams of heroin per day if you had the A1 minus status. So even something simple like this, where we're trying to look at predictors of outcome using our genetic marker, we've been able to see that over time, we've been able to predict response. We've undertaken a more sophisticated attempt at that too, looking at a randomised controlled trial, which we published in Nature Medicine in 1995, based on a couple of studies, one in Norway by Borg and one in Canada by Dongier, where they prescribed bromocriptine, a D2 agonist, in the treatment of alcohol dependence. There were 83 in this initial study. It was with 52 who completed the study and were prescribed 7 milligrams of bromocriptine a day. The overwhelming conclusion from this work is that it was the bromocriptine A1 group that had the biggest reduction in craving. Now, these were inpatients, and so that craving was an analogue measure of substance use, but we certainly found that for the 6 weeks that these individuals were in an inpatient setting, that their craving, if they had the A1 version of this gene and were administered a D2 agonist, bromocriptine, had the best effect. And then in terms of anxiety scores as well, we saw a similar effect, but it occurred over a faster time course between 0 and 3 weeks with the biggest changes. But it certainly intrigued us, and since then there have been some analogue studies, one looking at brain function with bromocriptine reducing impulsivity and changing activation in several key brain structures in a very similar way to the finding of Melanie White's study I described earlier. And there's been some important work looking at animal studies changing the preference of rats from alcohol to water, for example, on the treatment of bromocriptine, but no broader results in terms of larger clinical trials. And in fact, no marriage of this work with other forms of treatment, which we thought might be useful too, that if you lowered craving, it might make you more receptive to other treatments, but that integrated care hasn't been evident in studies yet. What we are interested in progressing in the next little while is a better understanding of complex cases that are hard to treat, and that's particularly with post-traumatic stress disorder. This study of cognitive behaviour therapy that we published in 2003 of 608 participants across Australia, we were very keen to look at the interaction between PTSD and alcohol use. And in particular, what we saw was that alcohol use at baseline wasn't predictive of outcome. So if you could treat alcohol misuse effectively and reduce the drinking in these people with PTSD, that they had much better treatment outcomes. And that was associated with less avoidance, numbing and arousal. But if alcohol use stayed high, the PTSD outcomes were significantly poorer. We've been very keen to consider what genetic factors might help to disentangle some of these issues, particularly given the challenges of combat, where those that are in combat are repeatedly exposed to stressors. And this is a review that we published last year, just showing in terms of the review, I won't go through any of these genes in particular, but the ANK1's the one that we've been talking about, the D2 dopamine receptor gene. There's 5 studies that look at PTSD and comorbidity, and some of them have been conducted by us. So this study here of 91 male Vietnam veterans with PTSD, all reported exceeding 60 grams of alcohol per day when off duty in Vietnam. 41.8% exceeded 60 grams per day currently when coming onto the study. And we were very keen to look at the relationship between their A1 allele status and their alcohol consumption. And this figure illustrates that. So clearly, of course, you'd expect the harmful drinkers in the sample to be drinking more than the non-harmful drinkers. That's a trivial graph. But when you separate them out in terms of those with the riskier genetic status for the DRD2, they're consuming on average about 130 grams of alcohol a day, and those with the A1 minus about 50 grams of alcohol per day. And when we looked at rates, so this is the average in grams per hour, it's really marked as well. So with 55 grams per average per hour for those with the A1 allele, and about half of that for those with the A1 minus status. And so we subsequently thought to develop, and this was a study that Bruce oversaw, the use of SSRI antidepressants in this group, to look at the interaction between, and this was an open label trial, between their genetic status and their response in particular to paroxetine, which we know induces D2 messenger RNA and increases the synthesis of D2 dopamine receptors. And I won't go through the sample in much detail, except to show in this open label trial of paroxetine, what we saw was the difference was here in social dysfunction. These are General Health Questionnaire 28 scores. And once again, this gave a very useful parallel where we got changes in social dysfunction on the administration of paroxetine for the A1 plus patients only, in a similar confluence to the ratings that we saw for social pressure self-efficacy and the work that we've seen in animal studies previously. So there was a unity across these studies looking at social function and improved dopaminergic tone. And I'm not going to talk about this study except to say it's in progress currently at the Princess Alexandra Hospital, where we're bringing together a number of these elements, where we're looking at cognitive behaviour therapy. We're also looking at pharmacotherapy with naltrexone and acamprosate, but also genetic response to these treatments as well over time. And this is just showing at the moment we've recruited 178 patients, but that's ongoing. So in summary, the treatment implications of genetic work to date is limited, even though we have these desktop aids that we see in some other forms of medical intervention, such as cardiovascular disease, and there's very little evidence that there's pharmacogenetic standalone approaches in addiction. But we think that integrating genetics, pharmacotherapy and psychological therapies in the next stage is going to be very important indeed. And lastly, I'd just like to say a couple of words about epigenetics, which I think is the window in terms of a more interdisciplinary integrated view of addiction. And epigenetics, the emergence of this really arose out of some really fine work that had been done in Spain that looked at the development of twins over time and the fact that the methylation of DNA in these identical twins was increasing with age. And so this was a way of an interaction with genetic status and the environment to improve and finally tune the expression of DNA through one of two processes. DNA methylation, where a methyl group is added to a cytosine guanine dinucleotide, and that means that it's more tightly bound and less likely to be transcribed, or histone modification, where an acetyl group's added to a histone group in the tail, and that also makes transcription more difficult. So these are the tails on the histone groups that can become more tightly bound with histone modification, and this is a cartoon just showing the methyl groups being added as well, which also affects the hydrophobic nature of the DNA and means it's less likely to be transcribed. Now, there are enormous bodies of work being invested in epigenetics worldwide at the moment, including this NIH Roadmap Epigenomics project, where they're looking at 111 reference human epigenomes that begin to map this methylation or histone modification across a number of different tissues. And as part of this, there's also a psychiatric genomics consortium for PTSD in particular, with 10,000 cases and 30,000 trauma-exposed controls. So these are large-scale studies trying to understand the interface between environment and genes, because the things that may cause methylation or histone modification to occur are a whole range of triggers, things like stress, things like being in an urban environment where you're exposed to a lot of violence. They may be chemical, but looking broadly at a whole range of these tissues. So there's going to be a tissue bank looking at either histone modification or methylation in these tissues, and then developing networks that link tissues, gene activity, and the expression of those genes to understand disease is going to be vitally important. And we also see this now in PTSD, linking early exposure to stress and subsequent manifestation of this stress in adulthood. This study here looks at FKBP5, which is a glucocorticoid receptor regulating co-chaperone and relates to the sensitivity of your stress system. Importantly, what this shows is that methylation or demethylation of this gene mediates the interaction between exposure to childhood trauma and your adult trauma reaction. So being reared in a stressful environment that causes changes in methylation to this specific gene appears to have profound consequences in terms of your adult life. And when we're beginning to build on this, we also see some of the potential mediators might express through to physical disease as well. This one looking at methylation of a number of different genes related to inflammatory processes in PTSD that also might compromise your physical health. And once again, there's these complex maps of all of the different methylation patterns and then looking at references, and this is hemostasis and wound healing are the biggest differences that occur up here in terms of this methylation. In addiction, we haven't progressed this much at all. Most of it's been with smoking because of the direct chemical impacts that smoking have on DNA methylation. But we're hoping to progress that ourselves using gene chips such as this one. This is the Illumina psycharray chip, which we've used for the first time this year. It only became available in June. But it looks at 271,000 markers, which we can look at simultaneously. And there's a similar human methylation chip as well. So what we're doing at the next stage is trying to understand through cohorts, such as those with PTSD, how to integrate this methylation data, this genetic data, with unfolding symptoms over time. And we've also done this with schizophrenia, and I've run out of time, so I won't talk about this in much detail at all, except to say that we were the first group in the world last year to study brain tissue and methylation in brain tissue and begin to understand in schizophrenia what these differences in methylation might mean. In this case, in this panel, these differences relate to neuronal survival genes, neurotoxicity genes, methylation genes, and embryonic development genes. But importantly, when we were drilling down into subgroups with schizophrenia, our old friend the DRD2 popped up as well. So we're very interested then to look at comorbidity, methylation, and epigenetics at the next stage of this analysis. So epigenetic models are going to be really vital in terms of the next stage of understanding addiction, because drugs themselves may produce epigenetic changes, but we certainly know from the work in PTSD that environmental stress does. And the final really point to bring to your attention on this basis is that these epigenetic models, because they embrace nature and nurture, also behove us to look at some of the environmental and social policies that we've got that may influence the gene expression of some of these genes that I've mentioned. Epigenetic effects don't imply biologically based interventions. Public health initiatives to increase safety in childhood, to produce robust psychological skills at a population level, to ensure effective social support and a more adaptive and caring community may be the best epigenetic interventions we have at our disposal. And so maybe Gwyneth Paltrow's right, that there may be a role for kale and wild blueberries after all. Thank you.